Good evening, all. Welcome back to the 23rd session of PSI online lecture series organized by Education Committee of Physiological Society of India. Myself, Dr. Shutapa Dash, Assistant Professor and Head, Department of Physiology, Rani Indira Devi Government Girls College, Jhargram, West Bengal, India. As we know that on fourth Saturday of every month since 2023, this program is organized, and this lecture series is mainly based on various topics of human physiology and related aspects. This initiative of PSI Education Committee mainly focuses undergraduate and master students of different fields like general students, medical students, paramedical students, and different other also as students associated with other fields too and associated fields too. With further delay, I want to introduce our today's speaker, Dr. Kaushik Bharati. Welcome, Dr. Bharati. It's my privilege to introduce you. Dr. Kaushik Bharti is a health policy consultant at UNESCO New Delhi and former consultant at World Health Organization. He obtained his PhD in physiology with specialization in vaccinology from the Calcutta School of Tropical Medicine, Calcutta, India. And he pursued his postdoctoral study in Liverpool School of Tropical Medicine, United Kingdom, specializing in immunology and molecular biology. He was formerly a scientist at the National Institute of Immunology, New Delhi, and a senior program officer at the Translational Health Science and Technology Institute, NCR, Delhi. He was also a visiting professor in the departments of physiology and microbiology at Vidyasagar University, Midnapur, West Bengal, India. Besides academics, he also has a rich consultancy experience, having worked for Global Health Strategies, New York, USA, Sanofi Pasteur, Lyon, France, Polysecure, Sydney, Australia, the Science Advisory Board, Virginia, USA, and the Initiative Against Diarrheal and Enteric Diseases in Asia, Lyon, France. His major research areas include virology, vaccinology, and public health. He has published 115 papers in reputed national and international journals and has also co-authored a book entitled Basic Concepts in Immunology. He has delivered numerous invited lectures at national and international conferences, including UK, USA, Thailand, Malaysia, Hong Kong, Singapore, Indonesia, South Korea, Philippines, and Pakistan. He is currently on the educational board of 33 journals from India, Sri Lanka, UK, USA, Canada, United Arab Emirates, and Pakistan. He is the editor-in-chief of the South Asian Association of Physiologists, SAP Bulletin, Colombo, Sri Lanka, and chairman of the SAP Research and Publication Subcommittee. He is also editor-in-chief of the Journal of Clinical Genetics and Genomics, Windsor, Berkshire, UK. He is on the peer review panel of 16 medical journals from India, UK, USA, Germany, Poland, the Netherlands, China, Indonesia, and Nigeria. He is also a PhD examiner of the University of Calcutta, India, and Lincoln University College, Malaysia. He is associated with 42 organizations across the globe, Notably, he is the Vice President of the Physiological Society of India, member of the Infectious Diseases Society of America, and Fellow of the Royal Society for the Public Health, London. He has received 16 awards for his research work from India, New Zealand, UK, and USA. Now, Dr. Bharti is going to emphasize today's topic, essential techniques in immunology. Sir, please welcome you again, and please, you are requested to start your session. Thank you, sir.
Thank you very much. First of all, I'd like to thank the PSI Education Committee for giving me this opportunity to deliver this talk. Especially, I'd like to mention Professor Prashanpriya Nayak, the President of the Physiological Society of India, Dr. Shampa Sarkar Bishas, the convener of the PSI Education Committee, and Dr. Sutapa Das, today's coordinator, for her nice introduction. Now, I will let me just present my slides. Uh, yeah, is my slides visible and am I audible?
But sir, make it full screen.
Yeah, it's full screen now. Can you see it?
Yes, sir. Yes, yes.
It's full screen. Okay. So, as you can see, I'll be talking about the essential techniques in immunology. So, the major ones that are of interest to us, but there are many others also, which I will not go into. And first of all, when we talk about immunology, the first thing that comes to mind is immunization. So, what is immunization? So, if you take an animal or a human and you inject an immunogen into it, it generates antibodies. So this is the basic thing, very simple, and this is a very simplistic approach that I'm saying, but it's much more than that. So when we talk about antibodies, we need to see how many classes there are. There are actually five classes based on the heavy chain of these antibodies. For example, IgG, it has a gamma chain. IgA has an alpha chain. IgM a mu chain. IgD a delta chain, and IgE an epsilon chain. And there are two types of light chain, either kappa or lambda, and there is only one type of light chain can be present in an antibody. Both types can't be present together. So suppose if this is a IgG molecule and this is the light chain. If this is a kappa light chain, this will also be kappa, and if this is a lambda light chain, this will also be a lambda light chain. And this is a typical IgG Y-shaped structure. This is a pentamer. This is the IgM, immunoglobulin M, and this is the first antibody that appears in the blood in the primary immune response, and this is the secondary antibody that appears in the secondary immune response. IgA is this is a monomer, and this is a dimer, and there is a J chain or joining chain, and this secretory component, and this is present in mucosal secretions. And epsilon chain is there in case of IgE. This is responsible for allergic reactions, and this is IgD. It has several functions. One of the main ones is B cell maturation, among others. And you can see the subclasses. IgG has four subclasses, IgG 1 to 4, with gamma chains, four types, and either kappa or lambda chain. In case of IgM, IgD, and IgE, there are only one type, and in case of IgA, there are two types. So these are the subclasses of the antibodies. Now, if we look at the typical structure of an antibody, it has a Y-shaped structure like this, and this is the heavy chain here in the inside, and this is the light chain. So, as the name suggests, this is heavier in in molecular weight, and this is lighter. And this at the very tip, this is the variable domains, because the composition of the amino acid changes, it mutates, and these are the this is C1 domain, this is C2 domain, and this is C3 domain. And at this carboxyl terminal, you have the FC portion. This is the fragment that is crystallizable, and this is a hinge region, that is it can fold, move back and forth, and these are the disulfide bonds. One thing that should be remembered is that only 2 to 5% of IgG molecules are antigen specific. So the majority, 95 to 98% of IgGs are non-specific. So there is a need to purify them and to concentrate them. So how do we purify antibodies? So we can use two types of strategies. One is low specificity. This includes the physicochemical approaches. And secondly, we can use high specificity approaches, that is affinity chromatography. Let's have a look at the physicochemical methods. So if we take a solution of protein and we add increasing concentrations of salt in the form of ammonium sulfate, and as we increase the concentration, what happens is that this protein precipitates out. So since it comes out of the salt solution, from the solution, this is called salting out. The exact opposite occurs when we decrease the concentration. As we slowly decrease the concentration of the salt, ammonium sulfate, this precipitate slowly goes into solution. So it again goes into solution. That's why this is called salting in. So this is the basic principle of physicochemical separation. Now let's have a look at size exclusion chromatography. This is another type of physicochemical method. First, you take a column with beads inside it. So beads may be dextran like Cephalodex. It may be Biogel A, which includes agarose beads, or Biogel P, which contains polyacrylamide beads. And when you put the mixture of proteins, large and small, what happens is as they flow through, the smaller ones enter into the beads, and that's why they get slowed down, whereas the larger ones pass through the spaces and they come out first. Now, due to their large size, these larger molecules are excluded from entering into into the beads. That's why, since it is due to the size why they are excluded, this procedure is called size exclusion chromatography. If we look at the actual photograph, you can see here when you add those samples, first, they separate out, and you get the larger molecules coming out first, the larger proteins. Then you get the wash buffer, and later on, the smaller molecules emerge like this. This is another physicochemical method, dialysis. Here we use a membrane, a dialysis bag, we sometimes call it, and it has pores on its walls, by which it differentially passes molecules. So this is a differentially permeable membrane, and there is a phenomenon. There's a term called MWCO, or molecular weight cutoff, by which we can regulate the particle size of the particles passing through the pores of this selectively permeable membrane. Suppose if the MWCO is 140 kilodaltons, then those protein molecules that are larger than 140 kilodaltons, they remain inside the bag, whereas those which are smaller than 140 kilodaltons, they diffuse outwards. And this is a passive diffusion method, so you have to keep on stirring using a magnetic stirrer, and take out the solution from outside intermittently, so that the molecules can equilibrate on both sides of the membrane. So this is the basic principle of dialysis. Now let's look at the affinity methods. Basic principle is that you take these beads are there, and to which this desired protein binds, they have affinity for that. That's why it's called affinity chromatography. Whereas other proteins that do not have affinity, they pass through. So these are non-binding proteins, and this is the yellow is the binding resin. So if you look at this, there are antigens on the surface which have affinity for these antibodies, and they bind to these beads like this, and this is called loading. When you load, absorbance is low at that time, and then when you wash them, when the unbound proteins are washed, you get a plateauing effect in the absorbance. And when you start eluting these, the target molecules, you get a sharp peak in this elution peak in this case. So we have a machine called a Biorad Econo Pump. This is manufactured by Biorad company. It's a basically peristaltic pump. So just like we have peristalsis in our gut, so similarly the fluid passes through the tubing in pulses. That's why it's like a peristaltic motion. There's a built-in this spectrophotometer inside where you can monitor the absorbance that is there, and you have a collecting tubes, many of them together, that you place at the back, where you can collect the wash buffer, and you can collect the eluted solution, all these things. So this is fully automated. It can be bidirectional. You can push it forward or backward. It is double channeled as well, so that you can mix buffers together. One may be concentrated, one may be dilute, so you can control the density of the buffer like that. So many functions are there, and this is a this is a uh, low. It's like the one we have, HPLC, high performance liquid chromo. This is a low performance, so smaller amounts of samples are purified. Then we have to check for the purity of the proteins. We use the SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. And this is, you can see here, the apparatus. Basic principle is that SDS, this is the sodium dodecyl sulfate, it has two functions. One is that it denatures the proteins, so that the, the tertiary structure is broken down, is just primary structure. The second function is that it imparts a uniform negative charge to the proteins. So what happens, that when the electrophoresis takes place, since there is uniform negative charge, only they migrate based on their molecular weight. And since they are negatively charged, they migrate from the negative pole, the cathode, towards the anode. And here in this case, the smaller proteins come out first, because this polyacrylamide, it acts like a sieve. And when these proteins are denatured, the smaller proteins, they're very small, like small types of small snakes, if you imagine them. So they can go through the sieve quicker, whereas the larger proteins which are denatured are longer. You can imagine them to be long snakes, and they have take time to migrate through that. And you can stain this with Chromacy Brilliant Blue R250, and it produces staining like this. And if you run a molecular weight marker, you can see the size. This one is around 45 kilodaltons. This is around 51 kilodaltons. This is around 64 kilodaltons. So then we have to do blotting to transfer the proteins that are present on the polyacrylamide gel onto membranes. So there are three types of blotting. One is Southern blotting. This was developed by EM Southern, and here we transfer DNA. And these two, Northern and Western blotting, they are named in order to match with the name of Southern blotting. They are not named after scientists. Scientists are different, but in this case, it is named after a scientist, EM Southern. And in case of northern blotting, we transfer RNA, and in western blotting, we transfer protein, which in this case is the antibodies. And the membrane that we transfer onto is the nitrocellulose membrane. And the buffer that is we use is the Tobin buffer, which is named after Harry Tobin, who developed this technique, invented Western blotting. And you can see here what we do is we take the polyacrylamide gel and we put the membrane beside it, and we put filter papers on either side, then sponges, and then we put it in a cassette. And when we pass the current, it travels from the negative end to the positive end. In the negative side is the gel, and in the positive end side, this is the membrane. So, why does it migrate again? You must remember that it underwent SDS page, so a negative charge was imparted onto the protein due to the presence of SDS, and that's why they proteins still remain negatively charged, so they pass from the cathode side to the anode side, and this is the direction of transfer. And the buffer that we use here, it's called the Tobin buffer, or sometimes it's also called tris glycine buffer. So it's composed of the tris base, glycine, methanol, and sometimes SDS is also used. So this is the buffer that is used, and you can detect the protein using Ponsu S strain. It is like a pinkish red type of stain, as you can see. So this is how you can detect the proteins. Now I'm going to describe two types of enzyme tagged assays. So one is ELISA and the other is ELISpot. So this is enzyme linked immunosorbent assay, ELISA, because an enzyme is used, and it's immunosorbent because it uses antibodies. So it was invented in 1971 by Peter Perlman and Eva Engvall. They are both Swedish scientists, and they both worked at the University of Stockholm. Peter Perlman was the lab director, the principal investigator, and Eva Engvall was the postdoctoral fellow. Peter Perlman passed away in 2005, but Eva Engvall still works at the University of Stockholm in the previous lab. So there are three formats of ELISA. One is direct ELISA. Here it is called direct because the enzyme is directly attached to the primary antibody, and it detects the antigen. So in both of this, direct and indirect, antigen is bound to uh, the substratum of that ELISA plate, and in this case, in the indirect, it's so called because the enzyme is tagged to the secondary antibody, so there is no direct contact. That's why it's called indirect assay. Substrate in all these cases is OPD, or orthophenyldiamine, and the enzyme is horseradish peroxidase, and it has nothing to do with horses or radishes. This is an enzyme found in horseradish plant which is used. And in case of this is called capture antibody. It is called capture antibody because it captures the antigen. It is also called sandwich assay because this antigen is sandwiched between the two antibodies, and this tertiary antibody contains the enzyme tag which produces the color. So the basic procedure, you add the antigens in this case, and they get coated on the bottom of the plate, ELISA plate, and then you have to wash it so that all the unbound antigens are washed away. Then you add the primary antibodies, again wash it thoroughly so that the unbound antibodies get washed away. Then you add the secondary antibodies, which are tagged to the HRP, or horseradish peroxidase, and then you again wash so that unbound antibodies are washed away. Then you add the substrate, OPD, and then you get a color development. Now, washing is of paramount importance in case of ELISA, because if there is any unbound, molecules, antibodies, or antigen, whatever, then it will block the absorbance. Because when you detect this, which I'll show you in the spectrophotometer, if there is a blockage, you will get a problem with the absorbance. You won't get the correct absorbance. And we generally use a multi-channel pipette, 8 channel pipette, so to speak, because The ELISA plate has is 8 by 12, so 96 wells are there. And if we use a 8-channel pipette, then we can add and take out uh, solutions, very easily and quickly. For washing, we use this type of wash bottle, and the wash buffer here is either PBST. PBS is phosphate buffered saline with an anionic detergent, that is Twin 20. You can also use TBST. This is Tris buffered saline with Twin 20. And nowadays you can get automated plate washers, just like many houses have dishwashers. In the labs, we can have automated washers also, which makes life much more easier for the scientists. So when you coat the plate with either an antigen or a capture antibody, so there is no color. But as soon as you add the substrate at the end of the assay, color starts to develop. You can see here yellowish color, and if you leave that for around 20 minutes, full color development occurs. It becomes brownish type of color, and at that time point you have to stop the reaction. Now how do you do that? You add zero point 0.2 to zero point 0.5 to 2 molar H2SO4, that is sulfuric acid, and you need just around 50 microliters per well to stop the reaction. After stopping that reaction, you take the absorbance reading in a microplate reader like this. Now I'll come to another technique called centrifugation. Here it depends on two forces. One is the centripetal force, which goes inwards, and then is the centrifugal force, which goes outwards. So in centrifugation, we are concerned with the centrifugal force mainly, and there are various types of centrifuges. You can have these types of tabletop centrifuges, medium size. Here you can accommodate these types of Eppendorf tubes of 1.5 ml to 2 ml capacities. In these types of microfuges, you can accommodate Eppendorf tubes about zero point 0.5 ml capacity. You can also have these types where you can accommodate 15 ml Falcon tubes, or larger ones where you can accommodate the 15 ml Falcon tubes also. And the maximum speed is around 14,000 RPM in case of these tabletop centrifuges. Now I'll discuss another one, a type of centrifugation. This is called density gradient centrifugation. Here we need to create a gradient of varying density. So we can use sucrose. This is called sucrose density gradient centrifugation. We add the more concentrated sucrose at the bottom. This is, you can take this as 20%. Then we add some more, around 10%, and at the top we add very dilute, that is around 5% sucrose. And when you spin this down, these form this type of gradient. And when these molecules, for example DNA is shown here, these molecules settle down in according to their own density. Like if the heavy molecules, they go to the bottom, the medium sized one in the center, and the small sized one near the top. So this is density gradient centrifugation. This is ultracentrifugation. It's the same principle as the tabletops, centrifuges, except that it's a very fast, like 150,000 RPM. Heat is naturally developed inside, so these are refrigerated. This is manufactured by Beckman Coulter. And these are the accessories. These are the rotors. This is a fixed rotor, it doesn't swivel. And this is a swinging rotor, it swivels. In the normal state it points downwards, but as it starts spinning it becomes horizontal. And these are the cassettes that you use in case of swinging rotor, and these are the tubes that you use in case of fixed rotors. Now, after the centrifugation, what you do, you have to take out the samples. So here, if suppose you want to take out the plasmids, you insert the syringe just below this layer and just aspirate, and it will come out. Likewise, for viruses, if you want to separate the capsids, that is the surface structure of the virus from the inner portions, then you can do it similarly. Nowadays, we use Ficol pack. This is a chemical that forms a gradient very automatically, actually. So, after taking out the Ficol pack, you put it into the tube and you overlay that with peripheral blood. So, we detect peripheral blood mononuclear cells. I was involved with developing a vaccine, a DNA vaccine for Japanese encephalitis virus, where we isolated the PBMCs. So by peripheral blood, we mean this blood is drawn from the veins. So they are in the periphery on the surface as opposed to the arteries which are deep seated. So we take out the peripheral blood and we overlay that with the overlay on top of the ficol, and then we spin it down, and after spinning we see the separation of the layers. At the bottom goes the red blood cells, this is the higher density, then a layer of ficol pack, and then on the top part you have the plasma, and in the intermediate layer you have the mononuclear cells, which you take out very carefully, inserting a automated pipette. It's a motorized pipette, actually, battery operated. Now, I'm going to discuss about another enzyme linked assay. This is similar in principle to the ELISA, except that spot formation occurs. This is called enzyme linked immunospot assay or ELISpot assay. Here we detect cytokines. During our vaccine development efforts, we wanted to determine what type of immune response was taking place. Was it Th1 or Th2? Depending on the cytokine profile, we can tell what type of immune response the vaccine is generating. So this was invented by Cecil Serzinski. He is a French scientist. He's now based in INSERM in Paris. INSERM is the French National Institutes of Health and Medical Research in Paris. When I met him, this was in 2011 at the International Vaccine Institute in Seoul. He was then the Deputy Director General there, and now at INSERM he is the director actually there. And this is his paper, famous paper on ELISpot, published in the Journal of Immunological Methods, which is a very prestigious journal. Now, the basic principle of ELISpot is that you use antibodies. In this case, it has to be a monoclonal antibody because each of the monoclonal antibodies have to be of a particular specificity. For example, this one colored red, this has affinity for interferon gamma, whereas this one in orange has affinity for IL-4. And the other format is similar to ELISA. For example, you can use a fluorescent method or a color method with primary and secondary antibodies, and these cytokines are released from the T cells, which we detect. If we look at the procedure, we can see first we coat the with the capture antibody, then we add the cells. These are the cells that we isolated, that is the PBMCs, and we add them. And in each step, we have to wash it properly, just as in case of ELISA, and these release cytokines which bind to these capture antibodies, and you detect it with the antibody, primary and secondary antibody, and you get spots. And if you observe these spots, these look like this, and we detect it using this type of microscope. This is a robotic microscope with a robotic stage along with the computer you know monitor, and this is manufactured by Zeiss. We use this very expensive microscope. It costs around 50 lakhs rupees in Indian currency, and here you actually have to train the computer to tell between teach the computer which is an actual spot and which is an artifact. So nowadays this life is much more easier. AI and machine learning is used for this, but at that time we had to use manual teaching methods. And this is the spots, and these are the artifacts that I was talking about. And the membrane that is used in case of ELISpot, there is no membrane. It binds to the bottom of the plate. But in this case, there is a membrane. It's called PVDF. It's polyvinylidene difluoride, PVDF membrane, that is used in case of LS spot. And this is the positive control that we use. We stimulate the T cells with concanavalin A. This is actually a plant lectin isolated from jack beans. It's a plant, and it has high affinity, especially for mannose and glucose residues on the surface of cells. And it non specifically stimulates the cells so that you get a huge number of spots. And this is the experimental. We were developing a vaccine for Japanese encephalitis virus. So in case of this, we use the envelope protein, E protein, to stimulate the cells, and you can see intermediate number of spots. And this is the negative control using RPMI media, very less number of spots. And RPMI is the Roswell Park Memorial Institute. This is where the buffer, the medium was originally produced. This was established by Ros, Roswell Park, a surgeon, in 1898, and this is name has been changed to Roswell Park Comprehensive Cancer Center, located in the city of Buffalo in the New York State. So by comparing these number of spots, you can say what type of response is taking place, how what is the strength of the response and the cytokine profile. Now I'll describe a few fluorescent tag based assays. First, I'll talk about immunofluorescence. So, the basic principle of fluorescence is that first, there is a nucleus in the center of the atom, and there are at least seven shells with the lowest energy nearest to the nucleus. There is K, L, M, N, O, P, Q, seven shells with increasing energy level. And when there is a electron, when it gets excited, it can move from a lower state energy shell to a higher energy shell. And when it again starts to come down to the ground state in an exciting form, it releases the excess energy in the form of photons. So this is the very basic of uh, fluorescence. Uh, and there is a phenomenon called Stokes shift. This is developed. The principle was developed by Sir George Stokes. He was an Irish mathematician and physicist actually, and he was associated with the royal family. First Baron he was, and he his alma mater was the University of Cambridge, as and he worked there all his life also. And in this Stokes shift, once the it is excited, this is the maximum of the wavelength at excitation, and this is the maximum wavelength in case of emission. So between excitation and emission, there is a shift in the wavelength. So this is called Stokes shift. And why I'm describing this is that this is a cardinal feature of the phenomenon of fluorescence. So there are two types of immunofluorescence. One is direct, where the enzyme is attached directly to the protein, and the second is indirect, where this is attached indirectly. Now you can stain with these fluorophores, for example, fluorescent isothiocyanate. Here is the hepatic cells, liver cells. You can see greenish fluorescence. You can get like this. You can also get red uh, color if you use tetramethyl rhodamine isothionates or TRITC. These are the cytoskeletal of the cells. And in case of GFP or green fluorescent protein, as you can imagine, this is obviously green color. And this is a typical fluorescence microscope, and it depends on the principle of fluorescence. So it is you put the your cells here in this on the stage. Fluorophores are added, and it fluorescence is picked up, and the emission, there are filters also, and that block out light that are not emitted, but they only pick up the emitted light emission spectrum, and you can observe that directly or take photographs. So this is how it essentially functions. Now I'll talk a little bit about flow cytometry. This depends on the principle, Coulter principle. It was developed by Wallace Coulter. He was an American electrical engineer, and he had 85 patents, of which the most famous patent, most important, is that which dealt with the Coulter principle. Here you use an electric field for counting and sizing suspensions of cells in conducting fluids. For example, if you have this type of setup, you have a container in which you have electrolyte, and you have a container inside that is a chamber, and with a very minute aperture, and on the outside you have the anode, in the inside you have cathode. Now, when cells pass through this after running the electric current, then this aperture is so small that it only allows one cell to pass through at a time, and this acts as an insulator, the cells themselves. So there is a fluctuation in the resistance which is detected here, and this fluctuation means that the number of cells that are passing. And since one cell can pass through, you can count each of them, and this is now automated actually. This is a Coulter counter. It used to be used for counting, doing the CBC counts, that is complete blood counts. Nowadays we have much more sophisticated instruments. Now I'll go into the fluorescence activated cell sorting, or FACS. So the basic principle is that you have a mixture of cells, and you pass it through a laser beam, and it takes up you know uh, emissions from and multiplies them in a photomultiplier, and these particles then pass through a nozzle actually, and before release the charge is imparted to them. Sometimes they may be positive charge, they may be negatively charged, or they may be empty, that is neutral. These positive charged particles, they travel towards a negatively charged electrostatic plate, and they are collected here, and the positive one, the negatively charged, goes this one towards the positive side, collected here, and the neutral ones are collected here. So this is the basic principle, very simplistic actually. And here you can see one of the latest types of cell sorters that are manufactured by Becton Dickinson, and this is called the BD Fax Discover S8 cell sorter. It is a very sophisticated machine, and it has many functions. Firstly, it is totally automated, the setup, sorting, workflow, everything. It has a staggering 86 detectors, of which 6 of them are imaging detectors. There are 5 lasers that can detect blue, red, violet, yellow, green light, as well as ultraviolet rays. And you can do 6-way sorting with this. And it has the latest software. This is called the BD Fax Codus software, and it has 3 sizes of nozzles. I told you that it comes out of a nozzle. So here you can have 3 types: 85, 100, and 130 microns, depending on the types of cell you want to isolate and size. You can use these. Now I am coming to the end. I'll just finish off with monoclonal antibodies, which are developed using hybridoma technology. Now, this technique, hybridoma technology, was developed by 2 scientists. One was George Kohler, he was a German scientist, and Caesar Milstein, he was from Argentina. And they shared the 1984 Nobel Prize in Physiology or Medicine with Niels Kajan. And their Nobel citation indicates that They made this discovery for their theories concerning the specificity in development and control of the immune system, and discovery of the principle and production of monoclonal antibodies. So this first part was done by Niels Kajan. So he studied the specificity of the uh, various antibodies, that is, they can recognize self or non-self, based on which you can get various types of autoimmune disease due to breakage of immunol tolerance. He also studied the structure of antibodies, based on which Kohler and Milstein developed the monoclonal antibodies, which I'll discuss here. So, why are they so called monoclonal? Because they are produced from a single hybridoma clone. So you have to fuse these spleen cells with myeloma cells to produce a fusion cell. This is called a hybridoma. It's a hybrid between B cells isolated from spleen and myeloma cells, which is a cancer cell of plasma cells. And you take just only one clone and you produce the monoclonal antibodies. That's why they're called monoclonal antibodies. They're produced from a single clone. So they're produced first by injecting an antigen into a mouse. Then you isolate the B cells. These are the antibody producing cells, and you take tumor cells, that is specifically myeloma cells, because myeloma cells are cancerous B cells. So why do we take this? Because this is also a plasma cell, and this is also, B cells differentiate into plasma cells during secretion of antibodies. So these are similar types of cells having a similar genetic origin. So they are fused. That's why they are taken. So they these hybridomas, they have inherited the immortality of tumor cells because they can divide indefinitely, and they have inherited the antibody producing property from these B cells. That's why they remain intact, and they are then expanded and produce the monoclonal antibodies. So you take the splenocytes and you take the myeloma cells and you fuse them with polyethylene glycol. It's like a glue that fuses the cells together, and this fusion is very, very critical. I've been involved in developing monoclonal antibodies, and I have seen that these fused cells destabilize very easily, actually. So it's very difficult to get the hybridomas, and you select the hybridoma cells by putting them in HAT medium. HAT contains hypoxanthine, aminopterin, and thymidine. And in this medium, only the hybridoma cells survive. Any unfused splenocyte or unfused myeloma cells die. So then you have to expand these hybridoma cells. Either you can go for in vitro amplification in a tissue culture flask, or you can inject it into mice into the peritoneal cavity. It forms ascites. These are pouches in between the abdominal wall and the abdominal organs. And in this ascites fluid are the rich monoclonal antibodies. And in either case, you harvest them and you purify them also. And then like I showed you in the initial slides, so we have come a full circle, so to speak, starting from purification to monoclonal antibodies, which again have to be purified. So I'll just finish off with some therapeutic applications. They can be used for inflammatory diseases like Crohn's disease or ulcerative colitis. Crohn's disease is an inflammatory disease where patches of inflammation can occur not only in the large intestine but also in the small intestine. Whereas in ulcerative colitis, you get inflammation primarily in the descending colon, the sigmoid colon, the rectum, and the anus, this region. So, in order to inhibit the inflammation, you can use two monoclonal antibodies, infliximab or adalimumab, which inhibit TNF alpha. So it causes inflammation. Now, any drug in which you see MAB at the end, you will easily know that this is a monoclonal antibody. All monoclonal antibody drugs end with MAB. MAB stands for monoclonal antibody. And you can use these two same monoclonal antibodies for treating rheumatoid arthritis, these debilitating, malformations that you get in the joints, in the hands particularly. These can be slowed down. In case of IgE, in the beginning slides I showed you, it's responsible for allergic reactions. Here, in case of allergic asthma, you can treat this with omalizumab, this IgE inhibitor, and also in case of basilizumab, you can inhibit IL-2, which prevents activation of T cells. So activated T cells can actually cause kidney transplant rejection. So this can be prevented with this monoclonal antibody. You can use monoclonal antibodies in case of cardiovascular diseases. Suppose there an coronary angioplasty needs to be done, where you can use balloon angioplasty to unblock the coronary arteries. There, coagulation can occur, and platelet aggregation can be the cause of this. You can use this type of platelet aggregation inhibitor, abicizumab. It inhibits platelet aggregation so that coronary angioplasty operation can go on smoothly. In case of infectious diseases also, like for example respiratory syncytial virus, RSV infection, it this RSV fuses with cell through the fusion protein, and this fusion protein adherence to the cell can be blocked by palivizumab, this monoclonal antibody which inhibits the fusion. So if the fusion protein cannot fuse with the cells, lung cells, then the virus can't enter the cells, so infection is halted. So this is another useful application. And last but not least, cancer therapy. This is especially a field where monoclonal antibodies are increasingly being used. You can use naked monoclonal antibodies. For example, CDC, this is complement dependent cytotoxicity, where you harness the power of the complement system. You can also use ADCC, this is antibody dependent cellular cytotoxicity. You can use other types of immunoconjugates. For example, you can conjugate a radionuclide with the monoclonal antibody. You can use a immunocytokine by conjugating a cytokine with the monoclonal antibodies, or a toxin to produce an immunotoxin. You can also use a part of a antibody, not the whole. This is called the single chain variable fragment, where one of the chain of of the light and one of the heavy chains are fused together with the protein, with the amino acid linker, and to that an enzyme is attached. And this treatment is called antibody directed enzyme prodrug therapy, ADEPT. So you attach this enzyme conjugate, and the cancer specific antigens that are present on the surface of the tumor cells, they are recognized by the SCFV, and they attach to that, to which the enzyme is attached. And when the prodrug is injected, product is the uh, previous version of an active drug. So when the prodrug is injected and it reaches this vicinity, only in this vicinity the prodrug becomes converted into a cytotoxic drug, so that it directly kills the tumor cells. So this is very specific. It doesn't kill other cells. Liposomes also, these are lipid membrane enclosed, globules. So it's just like a cell membrane, lipid bilayer, in which cytotoxins can be inserted. Here also, you can use SCFBs to direct them. You can also use natural killer cells bound to antibodies, and also you can use multi-step targeting like these using linkers. So I'll stop here. Uh, I have tried to describe the major types of immunological techniques, although I'm sure you know there are many others, and maybe sometime later, if I get the opportunity, I'll discuss about them. And I thank you all for your attention. And I did much of my research here in Delhi for many years. So thank you very much.
I extend my hearty thanks to our honorable speaker for today, Dr. Kaushik Bharati, for a wonderful as well as enriched presentation. He stated elaborately various immunological techniques. Starting from immunoglobulin classification, Dr. Bharati has nicely explained various techniques of antibody purification depending on specific strategies. Affinity methods, chromatography, SDS page, blotting techniques, ELISA, ELISpot, density gradient centrifugation, ultracentrifugation, immunofluorescence, different staining procedures like FITC staining methods, TRITC staining, fluorescence microscope, flow cytometry, counter instruments, FACS techniques, hybridoma technology, therapeutic applications of monoclonal antibodies for treating so many diseases like inflammatory diseases, cardiovascular diseases, and even cancer therapy. It seems that this lecture covers a broad field of essential techniques in immunology. Now I will ask audience if there is any questions. Please feel free to ask so. Is there any question from audience side? Yeah, there is one question from audience Orno Banerjee. The question is how can hybridoma technology produce monoclonal antibodies against low immunogenicity or self antigens? Dr.
This,
Bharati, can you hear me?
yeah, I can hear you. Uh, these hybridoma technology is currently still in its infancy, and these are mainly being produced against immunogenic cells, like I highlighted, for various diseases, and for self antigens, there is some problems are there, and in the from a technology standpoint, as I was highlighting, the fusion part, producing the hybridoma, is very difficult actually. As I highlighted, it's I have first hand experience regarding this, so that is difficult, and production is is hindered by these things.
Thank you, sir. Do you have any other questions from other audiences? Dr. Banerjee, have you more question?
Hybridoma technology cryopreservation can be used in case of what we do for cells, bacterial cells or mammalian cells, or ordinary antibodies. What we one thing that we can't do freeze thawing multiple times. So they are stable. If we put it in a, for example, we can put it in. These -80 freezers. We generally put it in aliquots, not more than 100 microliters aliquots separately, so that we can thaw it just once, then we discard it. Because if we freeze it, if it undergoes freeze thawing cycles, then the antibodies will degrade. So the same goes for in case of cryopreservation for ordinary antibodies as well as this monoclonal antibodies.
Okay, so sir have explained, Dr. Banerjee, the question you asked, sir. Uh, any other question? So I think this lecture definitely will encourage the students in future endeavors. I would also like to express our sincere thanks to Professor Proshun Priyanayak, President of PSI Education Committee, for his idea and initiative he taken for this lecture series and engaging other members of this committee for making it successfully. I must mention the name Dr. Shompa Sarkar Bishwas, convener of PSI Education Committee, for nice coordination and covering the whole program in very excellent way. I am thankful to the audience present here and present other PSI Education Committee members also for their enormous effort. On behalf of PSI Education Committee, I extend my sincere thanks to the Almighty for overall success of today's program. Here is another announcement. Our next series topic is clonal selection and immunomodulation, and the speaker is Dr. Shilpak Chatterjee, Principal Scientist and Assistant Professor, DBT Wellcome Trust Intermediate Fellow, Cancer Biology and Inflammatory Disaster, CSIR IICB Kolkata. So thank you again. Thank you, Dr. Bharti, again. Thank all again for your patient hearing. Good night, sir. Good night, all.